Hello, this is Bio 225, Genetics and Genomes, Chapter 3, Lecture number 3. We have already explored the Mendelian genetics in which we have an arrangement of one dominant and one recessive allele, and the dominant allele will always exhibit its phenotype, and the recessive phenotype will only be expressed if two recessives come together. Today, we'll continue our exploration into more complex allelic arrangements that are beyond basic dominant and recessive genes. There are cases in which the crossing of pure-bred lines can yield a progeny phenotypes that don't resemble either parent and therefore seem not to obey Mendel's rules. For example, let's look at lentils. They come in many colors and patterns. For some traits, it is observed that there is no definitive dominant or recessive gene. There may be more than two alleles that account for a phenotype. There may be multiple genes involved in an apparent phenotype altogether, and yet another variable may be gene-environment interactions. Last lecture, we looked at complete dominance, but dominance does not have to be complete. Dominance may be incomplete, as we see in snapdragon flower color as an example. Alleles may be codominant, such as in a lentil coat pattern or AB blood groups in humans. A gene may have more than two alleles, which is also true for lentil coat patterns, ABO blood groups in humans, and histocompatibility. Genes may be pleiotropic, meaning that one gene may contribute to several characteristics. For example, a recessive lethal gene in mice AY that also produces a dominant coat color phenotype. Usually, in this case, only the heterozygotes will carry the gene because the mice affected with double recessive AY gene die prematurely, where in some genes such allele can result in delayed lethality. Thus, the affected individual may live long enough to reproduce. Therefore, its progeny will be affected with lethal phenotype or will be a carrier of the gene. Now let's visualize complete, incomplete, and codominance. In case of complete dominance, hybrid resembles one of the two parents. What does it mean? If we have A1 allele for parent 1 and A2 allele for parent 2, and A1 is dominant over A2, then the offspring will have expression of A1 phenotype. If A2 is dominant over A1, then offspring will express A2 dominant phenotype. In case of incomplete dominance, we are having A1 and A2 alleles. However, offspring is having completely different, neither A1 nor A2 phenotype. In case of codominance, hybrid shows traits from both parents, which means it carries A1, expresses A1 and A2 phenotypes. Here is the snapdragon flower example for incomplete dominance. The parental alleles are white color and red color. However, the offspring expresses neither white nor red and has new color pink. Now, let's examine the incomplete dominance in a little bit more detail by looking at allele distribution of snapdragon flower color. In the snapdragon flower color, a heterozygous plant will not resemble any of its parental flower colors. This is because neither allele exhibits complete dominance over the other. So now let's look at the cross between a red flower with gene composition of big A, and a white flower with gene composition of little a. When these two plants are crossed together, all of their progeny, the F1, will be big A, small a. And in terms of phenotype, all of these plants will be pink. The interesting genetic composition seems further on in progeny F2. In F2, we see the reappearance of both red and white colors in roughly the ratio of 1 red to 2 pink to 1 white. This is because F2, which is a product of backcrossing of two F1 individuals, will have 1 to 2 to 1, big A to big A small to small a ratio. To study codominance, let's consider lentil coat patterns. In codominance, the F1 hybrids display traits of both parents. When we cross the spotted CS with the dotted CD parental plants, all of the F1 progeny are spotted and dotted CSCD. One parent donates CS and the other contributes CD. Therefore, the F1 progeny is CSCD. If the F1 is backcrossed here, the F2 progeny will produce the 1 to 2 to 1 ratio of 1 spotted CSCS, 2 spotted and dotted CSCD, and 1 dotted CDCD individuals. And just like in the incomplete dominance, the phenotype ratios reflect the genotype ratios. Again, in codominance, the F1 hybrids display traits of both parents. For example, AB blood group. The blood groups are characterized with sugar molecules being displayed on red blood cells. Let's consider that the gene I controls the type of sugar is on the red blood cells. Given that gene I has two alleles, IA and IB, there are three distinct individuals. IA individuals have A sugar. IB individuals have B sugar. IB individuals have both A and B sugars. Type of dominance depends on the type of proteins encoded and by the biochemical functions of the proteins. Variation in dominance relations do not negate Mendel's laws of segregation. Alleles still segregate randomly. Interpretation of phenotype-genotype relations is more complex. The gene is not restricted to only two alleles. However, individual can carry only two alleles. When we are talking about relationship of dominance and recessive, we are talking in relevance to one gene out of two. Now let's look at an example in which a gene has three alleles. In this case, it is the gene that determines the blood group, or otherwise known as A, B, O blood types. In this case, we have one allele that codes for sugar A, the second allele that codes for sugar B, and the third allele that codes for nothing. So when the genotype is AA, obviously our phenotype is an individual. This is said to have blood type A. Along the same line, if we have BB, our phenotype is an individual that is said to have blood type B. But in addition to A and B, we also have an I, which is an allele that codes for no sugar. Therefore, a genotype AI will also yield an individual with blood type A. Same thing holds true for genotype BI, also yields an individual with blood type B. When we see a genotype AB, that yields a phenotype that we call a person with blood type AB, which means that the red blood cells express both type A sugar and type B sugar. Please note that A and B are dominant over I and codominant with respect to each other. So, what are the medical implications of A-B-O blood group genetics? Our immune system can generate antibodies against molecules that are not natively found in the organism. So if a person has a blood type A, that means that he or she will have antibodies against sugar B, and vice versa. The blood type AB expresses both A and B sugars on red blood cells. Such individuals will not generate antibodies against A or B because both are natively expressed sugars. In case of individual with type O blood, this person will not express either A or B and therefore will generate antibodies against A and B. So why? A person with blood type A can only receive blood from another person with blood type A or a universal donor blood type O. This is because the individual with blood type A will have antibodies against type B sugar in their blood serum. So if accidentally B-type blood is introduced into such person, a very vigorous immune response against B antigen will be initiated. A person with blood type A can, however, receive blood from a person with blood type O, and that is because blood type O does not express either A or B sugar on the surface of red blood cells. So there is nothing on the red blood cells from blood type O donor for the antibodies to attach. Following the same logic, a person with blood type AB has no serum antibody for A or B antigen. Therefore, they can receive blood from either A, B, AB, or O blood groups, and otherwise known as universal recipients. Conversely, as blood type O is known as the universal donor. Now let's consider a gene with 5 alleles. Each allele may have its unique dominance correlation with other. In this case, we have gene C with 5 alleles for the gene: spotted CS, dotted CD, clear CC, marbled 1 CM1, and marbled 2 CM2. Reciprocal crosses between pairs of pure breeding lines is used to determine dominance relations. Figure 3.6. Reciprocal cross are crosses between different genetic stocks, strains, or species where the sexes of the parents are reversed. In this given example, we have gene for marbled pattern, CS, that dominance over others, and gene for spotted and dots that are co-express. Let's look at 3 examples from the previous slide. First one, crossing marble 1, CM1, CM1, crossed with clear CC, CC, will yield all F1 marbled 1, CM1, CC. F2 progeny yields 798 marbled 1, CM1, and 296 clear, CC. What does this mean? Is the ratio expected? Please explain. Example 2. Marbled 2, CM2, crossed with clear CC, produces all F1 marbled 2, CM2, CC. If in F2 progeny we get 1, 2, 3 marbled 1, CM2, and 46 clear CC individuals. What does this mean? Is the ratio expected? Please explain. Example number 3. Marbled 1, CM1, crossed with marbled 2, CM2, produces all F1 marbled 1. In F2 progeny, 272 individuals are marbled 1, CM1, and 72 marbled 2, CM2. What does this mean? Is the ratio expected? Please explain. 3:1 ratio in each cross indicates that different alleles of the same gene are involved. Dominance series, therefore, is as follows: CM1 greater than CM2 greater than CC. Now let's explore the human histocompatibility antigens, which are genes that are extreme example of multiple alleles. 3 major genes, HLA-A, HLA-B, and HLA-C, encode histocompatibility antigens. Histocompatibility is a protein complex 1, role of which is in differentiation between proteins that belong to the organism versus proteins that are of foreign origin, bacteria, viruses that need to be dealt with. Each gene has 20 to 100 alleles. Each allele is condominant to every other allele. Every genotype produces a distinct phenotype. Enormous phenotypic variation is where you try to calculate its up to 2 power of 100 different variations. Mutations are the source of new alleles. Mutations occurs whether being err in replication or under chemical or physical stress. These changes are not controlled and are usually determinate but not always lethal. If mutations occur in gamete-producing cells, they can be transmitted to offspring because egg and sperm will contain these mutations in their genomic DNA. Luckily, the rate of mutation is low, in the order of 10 power of minus 4 to 10 power of minus 6. This is partially because we have different proofreading mechanisms in our DNA replication that ensure no errors are introduced. Some mutations can result in phenotypic variants that can be used by geneticists to follow gene transmission. For example, we will explore the basis of mutations later in the course. Now we will introduce some common concepts and terminology. First, allele frequency is the percentage of the total number of gene copies for one allele in a population. Obviously, the most common allele is usually the wild type, plus allele. Rare allele is considered a mutant allele. It's safe to assume this. Second, monomorphic gene. Gene with only one common wild-type allele. Example is agouti gene. It is a gene that controls the distribution of the natural pigment, melanin, in the hair of mammals and helps determine their coat color patterns. Third, polymorphic gene. Gene with more than one common allele. High-frequency alleles of polymorphic genes are common variants. Extreme example, 92 plant incompatibility alleles. As we have already discussed, wild-type agouti allele A produces yellow and black pigment in hair. 14 different agouti alleles in lab mice, but only A allele in wild mice. Allele found in wild animal is known as wild-type allele. So laboratory mutations could be the following. Mutant alleles little a and little at. Little a recessive to big A. Little a has black only. Little at dominant to little a but recessive to big A. Little at mouse has black on back and yellow on belly, as you can see on this picture. One gene can also contribute to several characteristics. We have already introduced the term pleiotropy, which is the phenomenon of single gene determining several distinct and seemingly unrelated characteristics. For example, many Aboriginal Maori men have respiratory problems and are sterile. Defects due to mutations in a gene required for functions of cilia, failure to clear lungs, and flagella, immortal sperm. With some pleiotropic genes, heterozygotes can have a visible phenotype. Homozygotes can be inviable. A prime example is the AY allele of agouti gene in mice. A Y allele of agouti gene causes yellow hairs with no black. Crossing agouti and yellow mice yield 50-50 ratio of agouti and yellow. Therefore, we can assume that yellow must be heterozygous for A and AY, with AY is dominant to A, as can be determined by producing the F1 and F2 progeny, as we have already seen. Now let's look at extensions to Mendel's law. What Mendel described: complete dominance, two alleles, all alleles are equally viable, one gene determines one trait. Extension: incomplete dominance, codominance, multiple alleles, recessive lethal alleles, pleiotropy, where one gene influences several traits. Extension's effect on heterozygous phenotype. Unlike either homozygote, multiplicity of phenotypes, no effect, several traits affected in different ways depending on dominance relations. And finally, extension's effect on ratios resulting from an F1-F1 cross. Phenotypes coincide with genotypes in a ratio of 1 to 2 to 1, a series of 3 to 1 ratios, 2 to 1 instead of 3 to 1, different ratios depending on dominance relations for each affected trait. Let's examine another case of mutation: sickle cell anemia. It's a result of mutation in a single amino acid in hemoglobin. Hemoglobin transports oxygen and CO2 in red blood cells. It is composed of two subunits, alpha globin and beta globin. Mutations in beta globin gene cause beta thalassemia, disorder that reduces the production of hemoglobin. Most common mutation of beta globin, HB beta S, causes sickle cell disease. It's a single amino acid mutation, glutamine to valine. This gene is pleiotropic, affects more than one trait, which include deformed RBCs, anemia, heart failure, resistance to malaria. It could be recessive lethality, heart failure. It can express different dominant relations for different phenotypic aspects of sickle cell disease. Here is a figure showing the disease phenotype. Normal red blood cells have high oxygen binding capacity and are donut-shaped. Sickle cells have elongated morphology and significantly reduced oxygen binding capacity, therefore manifest as anemia. Interesting fact is that people with sickle cell anemia do not contract malaria because the parasite cannot travel through blood, therefore cannot reproduce. This is seen in both sickle gene carriers and affected individuals. Mendelian genetics can be expanded for multifactorial inheritance, which means that two genes can interact to determine one trait. Such interaction may bring about new phenotypes as a result of gene interactions. We have already seen such example with lentils. Gene actions can also be complementary, with the example that we saw with the flower color. We can also observe epistasis, which means that interaction of genes are not alleles. It is the suppression of the effect of one such gene by another. Examples: Bombay phenotype in humans, in which a second antigen produces O phenotype even when both parents do not carry O allele. In all of these cases, F2 phenotypes from dihybrid crosses are in a variation of the 9:3:3:1 ratio expected for independently assorting genes.